learned a lot about lipids, but there's been an elephant in the room that hasn't gotten the coverage it deserves. Hello, darkness, my old friend. It's cholesterol. Cholesterol is a precursor to other molecules like steroid hormones and the bile acids, but there's a lot more to say about the molecule that some consider the wicked witch of the West due to its association with coronary artery disease. The reputation isn't completely deserved, though. For example, it isn't cholesterol alone that's plugging arteries in atherosclerosis. The cholesterol is mostly riding along in a car containing some crooks on their way to a crime. We'll learn later in the lecture that the real criminals are a bit of a surprise. In this lecture, we'll talk about the cholesterol elephant and some of its offspring, such as the bile acids and steroid hormones. We'll look at the place the elephant spends most of its time in cellular membranes. And we'll investigate the possible crime scene as cholesterol moves around in lipoprotein complexes. Okay, so where does cholesterol come from? You might be surprised, but a good deal of our cholesterol is actually synthesized in the body and does not come from the diet. This is another example of you are what you make, not what you eat. This is why the control of cholesterol synthesis is a big factor in health. Understanding how cholesterol synthesis occurs also suggests strategies for reducing the body's cholesterol level. So, in the spirit of know your enemy or know your frenemy, let's look at the key steps in cholesterol synthesis. The first step in cholesterol synthesis starts with acetyl-CoA molecules. These are the same molecules used for fatty acid synthesis and obtained from the breakdown of numerous molecules. The pathway to cholesterol from acetyl-CoA is long, involving 28 steps, but many cause only very minor changes. Most of the action in the synthesis actually occurs in the first 9 reactions. Cholesterol synthesis pathway partly overlaps with that of ketone body synthesis through the joining of 3 acetyl-CoA molecules to make a 6-carbon molecule known as HMG-CoA. But at this point, the 2 paths diverge. A reaction catalyzed by an enzyme called HMG-CoA reductase converts HMG-CoA into mevalonate, and we're off to making cholesterol. If you want to manage production of cholesterol in the body, HMG-CoA reductase is the master switch you're looking for. It regulates the entire cholesterol synthesis pathway. The enzyme is controlled by several mechanisms. The most important one for us occurs when cholesterol binds to it and turns it off. Now, this is classic feedback inhibition, where the end product of a pathway binds to an enzyme and shuts it off. When this happens, the entire pathway is halted. No need to waste resources and energy when cholesterol is abundant. Since HMG-CoA reductase is central to cholesterol synthesis, it is the target of anti-cholesterol statin drugs like Lipitor. These are drugs also known as reductase inhibitors, and what they do is mimic the structure of HMG-CoA and bind to the enzyme in its place, thus acting as competitive inhibitors. On the path to making cholesterol, mevalonate gets decarboxylated to convert it into 2 slightly different 5-carbon molecules we call isoprenes. And we will use the shortened names of DPP and IPP for these isoprenes. The next several steps involve joining these isoprenes together. So if you connect an IPP to a DPP, you get a 10-carbon molecule called geranyl pyrophosphate. Adding another IPP makes a 15-carbon molecule called farnesyl pyrophosphate. It's a metabolic jumping-off point, providing alternative pathways to make coenzyme Q or heme. However, on the path to cholesterol, what happens next is a joining of 2 farnesyl pyrophosphates to make a 30-carbon molecule known as squalene. Now, squalene is interesting. It can be rearranged into a cousin of cholesterol called lanosterol. And this 30-carbon molecule goes through numerous minor alterations. There's actually 19 of them in total, before finally arriving at cholesterol. So, how about a song? Some things that you can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together 1, 2, 3, 6 carbons known as HMG, and you're on your way to mega cholesterol. To synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to mega cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly, 10 carbons in each geranyl in the cell's routines to mega cholesterol. A single step licks farnesyl's, but that's not all. The squalene rearranges to lanosterol. From that, there's 19 steps to go to make cholesterol. You know that's the protocol to mega cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the look of HMG-CoA, and they give us ways to not make cholesterol. However, making cholesterol, if we don't make too much, is really important, and not only for making membranes. Cholesterol is a precursor for synthesis of several molecules. The first is a class known as the bile acids. Bile acids answer the following question. If your body wants to dissolve up a fat in your digestive system, who are you going to call? Yep, bile acids. Bile acids are detergent-like lipids made from cholesterol by addition of polar groups, creating a much more amphiphilic molecule than cholesterol. The digestive system is a polar aqueous environment, and the detergent actions of the bile acids solubilize nonpolar molecules like fats, cholesterol, and fat-soluble vitamins. Bile acids and cholesterol are implicated in formation of gallstones. Up to 25 million people have them, but they may not know it until one blocks the bile duct carrying bile into the digestive system. Then a painful condition arises. Now, it may surprise you to learn that gallstones are mostly cholesterol. Remember that bile acids are synthesized in the body from cholesterol, so where there are bile acids, there's going to be cholesterol. If the cholesterol to bile acid ratio gets too high, then crystals of cholesterol form with painful results. Another group of molecules made from cholesterol is the steroid hormones. Although most infamous as a way to get yourself banned from athletic competitions, steroids do everything from balancing ions and controlling inflammation to acting as sex hormones. The starting reactions for all steroid hormones are additions of hydroxyl groups to cholesterol. The first steroid hormone made is called pregnenolone, which is the branch molecule for the synthesis of the 5 steroid hormone groups. These are the progestogens involved in pregnancy maintenance, the mineralocorticoids, which perform ion balance, the glucocorticoids, which have miscellaneous functions including roles in inflammation, the androgens, which are the male sex hormones, and the estrogens, which are the female sex hormones. Female hormones such as estrogen are made from androgen male hormones by action of an enzyme called aromatase. Aromatase is targeted by drugs called aromatase inhibitors, and they're used to block female hormone production, a strategy for stopping estrogen-stimulated cancers. Another class of steroid hormones called the glucocorticoids inhibits the enzyme that makes the arachidonic fatty acid available in cells. Arachidonic acid, you may remember, is the precursor for making painful prostaglandins. Glucocorticoids such as cortisone stop the release of arachidonic acid. With no free arachidonic acid available, there's no prostaglandin production and so no inflammatory response. Glucocorticoids are used to treat painful or dangerous swelling. Glucocorticoid drugs are also why you hear aspirin and ibuprofen referred to as non-steroidal anti-inflammatory drugs, or NSAIDs as they're called. This mouthful expression was concocted to distinguish them from the steroid drugs based on the glucocorticoids that also inhibit prostaglandin synthesis. Probably the single most important role for cholesterol is in cellular membranes. Its presence in membranes helps manage their fluidity. Now, the rigid planar structure of a cholesterol molecule fits nicely in the gaps between the hydrophobic tails of the fatty acids, making membranes stiffer at moderate temperatures. It also helps to significantly reduce the movement of small water-soluble molecules in and out of cells, thus maintaining membrane integrity and ensuring orderly movement of materials through protein transporters rather than randomly through the membrane. Now, at very low temperatures, cholesterol can insert itself between the lipid tails and keep them from packing together too closely, so it works at high and low temperatures. The lipid bilayer, thanks to cholesterol, is remarkable in protecting the cell. Though some molecules like water, carbon dioxide, oxygen, and carbon monoxide are allowed passage, most other molecules are blocked and only get in through specific gates or channels in the lipid membrane. So cholesterol helps cell membranes to exclude what we call nonspecific passage of most molecules through the lipid bilayer. That means that the bilayer is a barrier to the movement of most molecules. On the other hand, specific passage of molecules in and out of the cell is managed by gatekeeper proteins known as membrane proteins. Together, membranes and their protein gatekeepers control movement of materials in and out of cells. This control of transport determines everything from the vitality and fate of cells to the very beating of our hearts. Membranes not only create the outer boundaries of cells, they also define the inner compartments within cells, like nuclei and mitochondria in eukaryotic cells. The same basic rules for making cell membranes hold for making organelle membranes. The foundational molecules are glycerophospholipids and sphingolipids, as we have seen. There are some differences, though, in composition. For example, the mitochondrial membrane houses the proteins involved in ATP generation. Now, so important is this function that the inner mitochondrial membrane is packed with proteins, about 80 to 90% by weight, versus only 20% or so for lipids. The cell membrane, by contrast, only contains about 60% protein by weight. Mitochondrial membranes are rich also in a lipid not found elsewhere. It is known as cardiolipin. Now, damage to this lipid is part of the cellular signal for suicide when the mitochondrial function is impaired. In this process, a cellular peroxidase enzyme catalyzes an oxidation reaction on cardiolipin. That leads to membrane rearrangement and release of the protein cytochrome C. And this is what activates the suicide mechanism of cells. It's tempting to think of membranes as fixed, impermeable structures frozen in time, but the fact is that they must be fluid-like and semi-permeable to function for a living cell. Cells and their membranes are sort of like balloons filled with water. They're extremely flexible and can very easily change shape, but the membrane must be strong enough to avoid bursting and spilling its contents. Cells biochemically alter their own membranes to keep them fluid at the temperature of the organism. Much like the fats and oils in our kitchen have melting points, so it is that membranes containing fatty acids transition from a more fluid-like state to a more solid state as the temperature drops. It's sort of like a freezing point. For human cells at a relatively constant body temperature, this isn't too big of a deal. But it is for organisms like fish in icy waters. The biochemical adaptation involves changing the mix of fatty acids present in the glycerophospholipids and sphingolipids. Remember that unsaturated fatty acids have kinks in their hydrocarbon tails that keep them from packing too closely. Cells increase the unsaturated fatty acid content of membrane lipids to keep them fluid at lower temperatures. The high content of omega-3 polyunsaturated fatty acids in fish membranes helps them stay fluid in chilly temperatures. And yes, those are the same omega-3s found in cold water fish like salmon, mackerel, sardines, and the fish oil products like cod liver oil. Membranes also contain a fair amount of cholesterol, occupying 14% of the dry weight of brain tissue. Membranes are important for cellular barriers, but barriers can be a bane as well as a boon. Cells need nutrients such as glucose, which can't cross the membrane as is. So cells must have mechanisms for transporting needed molecules in and other molecules out. This is why cells have a vast array of membrane proteins, each of which is typically very specific for the molecules or ions they move. The importance of membrane proteins is also shown by their relative abundance. On average, about 50 to 60% of cellular membranes' weight is made up of membrane proteins. The other half are lipids, although because proteins are much heavier than lipids, this means that there are far more lipid molecules. Proteins in the lipid bilayer have numerous functions, but let's focus here on the protein gatekeepers that control material crossing the lipid bilayer. Consider glucose transporter proteins that we abbreviate as GLUTs. They're found in the membranes of almost all animal cells. GLUTs help cells to import glucose. Now, true to their name, glucose transporters only allow passage of glucose molecules. Specific transport of molecules through membranes is vital for life. A variable in moving molecules across a cell membrane is their relative concentration on either side of the barrier. If there's a higher concentration of a molecule outside the cell than inside, the molecule will tend to move into the cell. We saw this with protons moving through the ATP synthase in the mitochondrion, for example, thanks to the proton gradient. Glucose has a higher concentration outside of a red blood cell than it does in. So the glucose molecules move from outside to inside through the glut by a process of passive diffusion through the glut, also known as passive transport. Now, in the nephron cells of the kidney, however, movement of glucose is much more challenging. The kidneys, which filter waste and extra water out of blood to make urine, normally reabsorb glucose to prevent it from being excreted in the urine. Now, this can result in higher glucose concentrations inside the nephron cells than in the surrounding fluids. So glucose cannot simply diffuse into the cells through a passive single-purpose transporter like a glut. That's why these cells have a different kind of glucose transporter called the sodium glucose transport protein. This cotransporter solves the problem of getting glucose into the nephron cells by using the fact that there's a high concentration of sodium ions outside the cells. Because there are more sodium ions outside, they tend to enter the cell where there is less. So a way for glucose molecules to get in would be to piggyback into the cells with the sodium ions that are going in. Now, you might wonder how it is that the sodium ion concentration is higher outside of nephron cells than inside. Well, that turns out to be true for all animal cells, and there's a very good reason for it. Another transport protein moves sodium out of cells. The protein that pumps out sodium is a revolving door antiporter called the sodium-potassium ATPase. What it does is it uses ATP as an energy source to move two ions in opposite directions. Okay? 3 sodium ions are moved out of the revolving door as 2 potassium ions move in. The result is a higher sodium ion concentration outside the cell and a higher potassium ion concentration inside the cell. Now, thanks to the sodium concentration gradient, glucose can piggyback into kidney cells that already have a high concentration of glucose within them. And by the way, anytime a transport system moves at least one molecule against a concentration gradient, as is happening with glucose here, we refer to it as an active transport system. So both the sodium-glucose system and the sodium-potassium ATPase system meet these criteria. All normal cell membranes use the sodium-potassium active transport system. The sodium-potassium ATPase performs a crucial function in the heart. calcium is the stimulus for contraction of the heart, as it is in other muscles. After contraction, relaxation of the muscle requires reducing calcium ion concentration inside the cells against a gradient. One way to reduce calcium ion in the heart is through action of another antiporter called the sodium-calcium exchanger. Sodium-calcium exchange depends on the high sodium ion levels outside the cells built up by the sodium-potassium ATPase. The sodium-calcium antiporter functions by moving calcium ions out as sodium ions move into the cell. When this happens, the muscle relaxes and the contraction cycle is complete. Now, in people with congestive heart failure, the heart is unable to pump vigorously enough to meet the body's needs. So how to strengthen the heartbeat for such people so that they could safely climb a flight of stairs or avoid swelling? Well, one way is to increase the level of calcium in the heart cells. What about tweaking the sodium-calcium exchanger? You betcha. An easy way to increase calcium inside the heart cells is to reduce the activity of the sodium-calcium exchanger. Now, this can be easily done by reducing the concentration of sodium ions outside the heart cells. And how do we do that? Well, that's accomplished by reducing the action of the sodium-potassium ATPase. Lowered sodium ion concentrations outside the heart cell mean less active transport of calcium out of the heart cell by the sodium-calcium exchanger. So what would inhibit the sodium-potassium ATPase? Digitalin, also called digoxin, which is a molecule produced in foxglove plants that is a very potent inhibitor of the sodium-potassium ATPase found in heart muscle. Extracts from foxglove plants have actually been used as treatments for hundreds of years. Their effectiveness was proven in 1785. Thanks to foxglove, many people live better lives. And thanks to biochemistry, we know the molecular basis of its action, reducing the activity of the sodium-potassium ATPase in heart muscle. So here's the key idea to remember about membranes. Lipid bilayers provide cellular boundaries, and proteins are the gatekeepers. We call these proteins integral membrane proteins. Each of them also provides an opening or binding site specific for the molecules being transported across the membrane. Remember that membranes need cholesterol and cells need other lipids as well. In order for cholesterol to do its magic, or for fat to provide energy for hungry cells, these water-hating molecules must first overcome their hydrophobic urges and travel through the body in an organism that is mostly water. Now, doing this requires extra steps, and this is one of the reasons that fat and fatty acids are not used as rapid sources of energy. They're insoluble in water, so cholesterol and other lipids must be made water-soluble so that they can travel through their watery surroundings. So just like you would send a 10-page report through the mail with an enclosing envelope, so it is that cells package up lipids in the body. That process with dietary lipids begins in the digestive system when they encounter the aqueous environment of the stomach. Just as we use soap on our hands to make grease soluble in water, the digestive system's detergent-like bile acids prepare dietary fats, cholesterol, and other lipids for transport across the intestinal wall. This process, called emulsification, creates small droplets called micelles, which make the lipids water-soluble and therefore capable of being delivered to the intestinal cells. Once inside the intestinal cells, fats, together with other lipids, get packaged into protein phospholipid bundles we call lipoprotein complexes. The lipoprotein complexes made in intestinal cells are ultra-low density lipoproteins, known as Ldls, or also chylomicrons. And as you can see, these complexes are organized so as to hide the hydrophobic triglycerides in the interior of the structure, surrounded by phospholipids. The outside includes lipoproteins and phospholipids, both of which are water-soluble. So lipids are shielded from their aqueous surroundings by the water-loving proteins and phospholipids in the complex. In the body, dietary lipids begin their journey in chylomicrons by entering the lymphatic system, which empties into the bloodstream. There, enzymes known as lipoprotein lipases, which are released by cells, begin to digest the fats in the chylomicrons. As these fats break down to fatty acids and glycerol, other fats, though not cholesterol, may be released from the chylomicron to be absorbed by cells. The chylomicron shrinks considerably as it loses these components, and its reduced size allows it to move freely through the capillaries. This shrunken structure travels the bloodstream to the liver, where it gets absorbed and its contents are stashed away. Now, the liver is the major modulator of metabolites that the body needs. It's in the liver that blood glucose concentrations are managed, and it is also the liver that monitors and addresses the body's lipid needs. Absorbing chylomicron remnants is not the end of the liver's story, though. It must also release packaged up lipids to meet the body's needs. Let's assume the liver has just sensed that the body needs fat and cholesterol. When this happens, the liver begins creating some lipoprotein complexes of its own. These complexes are known as very low density lipoprotein complexes, or VLDLs. As the very low density name suggests, VLDLs are relatively fluffy complexes containing cholesterol and fats that travel in the bloodstream, much like the chylomicrons did. After VLDLs leave the liver and enter the bloodstream, lipases digest the fats, but not the cholesterol in them. And as a result, like we saw with the chylomicrons, the VLDLs shrink, becoming slightly higher density lipoproteins you rarely hear about called IDLs, and eventually low density lipoproteins, or LDLs. Now, LDLs are what your doctor refers to as bad cholesterol when you get the results of your annual checkup. What's so bad about LDLs? Well, LDLs contain the highest concentration of cholesterol of all the lipoprotein complexes. The problem isn't the cholesterol itself, but mostly the unsaturated fatty acids LDLs contain. Yep, you heard that right. Unsaturated fatty acids. Remember the things you can get from unsaturated fats that are supposed to be good for you? These are susceptible to oxidation, and this is the first step in the development of an atherosclerotic plaque. The higher the levels of LDLs, the greater the likelihood of damaging reactions with the unsaturated fatty acids. Oxidized LDLs are swallowed by immune cells called macrophages, triggering a process that ultimately results in a physical block called an atherosclerotic plaque in the blood vessel. So does this mean that you should only eat saturated fat or stop eating unsaturated fat? Nope, absolutely not. Numerous studies show that is even worse for you. But it's a reminder that there are no magic foods, and the interplay of diet and health is far more complex than supermarket magazines would have you believe. Atherosclerotic plaques can cause heart attacks by blocking blood flow through arteries supplying the heart. Plaques can also rupture and trigger clotting. In contrast to the blockage of coronary arteries, clots are free to move anywhere in the body. And if they lodge in a critical supplier of blood to the brain, a stroke can result. One subtype of LDL called pattern B is smaller than other LDLs and may be especially responsible for plaque formation. So if LDLs are so dangerous, why does the body make them? Well, LDLs are a means of delivering cholesterol directly into cells in a process called receptor-mediated endocytosis. Cells have proteins called Ldl receptors embedded in their plasma membranes, and these receptors stick out of the membrane and latch onto LDLs from the bloodstream. When LDL receptors bind to the LDLs, the cell membranes in the area forms a sort of bubble that swallows the receptors with their LDLs and bring them inside of the cell. Once inside the cell, the LDLs are released from the receptors and get digested by enzymes to free the cholesterol and lipids that the cell needs. So how does the liver know when the body needs lipids? Well, that mechanism is interesting. The liver keeps tabs on LDL levels in the bloodstream. When cells in the body are hungry for cholesterol, they take up lots of LDLs, and this causes LDL concentrations in the blood to drop. So the liver notes the drop in LDL levels, and when that happens, it ups the production of VLDLs until the LDL levels rise again. When all is working well, LDL levels stay in check, aside from these minor fluctuations that I've just described. Now, one problem arises when the liver's capacity to buffer dietary fat and cholesterol is overloaded, leading to high levels of LDLs in the blood. Now, this can result from a high intake of fats, particularly saturated fats like butter or lard, or trans fats such as those in vegetable shortening or margarine. This is why the first approach to tackling high LDL levels is by diet alteration. Exercise can also play a role, but when lifestyle changes don't reduce LDL levels sufficiently, drugs called statins are typically used. Statins inhibit the endogenous synthesis of cholesterol in the body, as we have seen. But LDL levels can be high even if your diet is low in fat. If you're unfortunate enough to have a mutation in your Ldl receptors that make them non-functional, then you're in trouble. If that happens, LDLs from the blood cannot be bound by the receptors and cannot be delivered into cells. As a consequence, high levels of LDL accumulate in the blood. And this is what happens to people who suffer from familial hypercholesterolemia. People with such mutations develop dangerously high levels of serum cholesterol as children, putting them at risk for atherosclerosis and coronary heart disease at an early age. But what about the so-called good cholesterol known as HDLs or high-density lipoproteins? What makes them good and why is it desirable to have lots of HDLs? Like the other lipoprotein complexes, HDLs are involved in movement of lipids, but unlike the others, levels of HDLs are inversely correlated with atherosclerotic events, meaning the more HDLs you have, the less likely you will get atherosclerosis. Why is this? Well, HDLs actually remove cholesterol from macrophages during the formation of atherosclerotic plaques, thus reducing the incidence of plaques. HDLs also take extra cholesterol from other cells back to the liver or to adrenal glands, testes, or ovaries where it is used to make steroid hormones. This reduces the concentration of free cholesterol in the body. Now, what's more, HDLs inhibit oxidation, inflammation, blood coagulation, and blood platelet aggregation, all of which are factors in coronary heart disease. Now, given all these good effects of HDLs, can we do anything to increase their levels? Yes, and it turns out to be what's good for you more generally. Exercise, weight loss, consumption of omega-3 fatty acids, increased intake of fiber, reduced carbohydrate intake, and removal of trans fats from your diet are all known to increase HDLs. The word cholesterol has become a catch-all. There's only one cholesterol molecule, but unfortunately, LDLs and HDLs have become known as bad cholesterol and good cholesterol, respectively, even though it's not the cholesterol that makes them bad or good. You'll even see the entire class of lipoprotein complexes referred to as total cholesterol, even though that includes the triglycerides of ordinary fat. This is a mess that is unlikely to get cleaned up soon. Meanwhile, the diverse molecules of membranes and lipid transport have been swept under the biochemical rug for too long. Cholesterol's important role in membranes has been obscured, but more importantly, it has been a victim by association, embedded in atherosclerotic plaque-forming LDLs. That's not to say you shouldn't reduce your unhealthy LDLs. You should, but it should be accompanied by other dietary and lifestyle changes too. There is still an awful lot we don't know about cholesterol, despite awarding 13 Nobel Prizes to scientists doing cholesterol research. The times they are a-changin', though, and new technological advances are exciting. Now, as scientists create a complete profile of all the lipids of a cell simultaneously in the science known as lipidomics, the mysteries and wonders of these diverse molecules will be revealed.